Very well. So, first of all, I thank everyone for joining our session, and we sincere thanks to the localization world as well to giving us a platform to speak on the process. Uh, it's a great knowledge sharing opportunity for everyone, as well as we get to interact with the industry experts in complex drug market space and equipment working. So my name is Rakesh Bhardwaj. I'm from I handle the operations and business for Truking India as a director. And with me online, we have our HQ team also joined from China as well, with the Doctor who holds a PhD in cellular immunotherapy from University of Pennsylvania, along with the process and marketing department on the standby support from there. So let us start the presentation, and we will also, in the end of the session, we would like to have question answers as well. In case of any doubts and further clarifications, we can do the coordination as well at later stage over the emails as well. Okay, let's get going. So a little bit brief about our organization. Truking Technology is a Chinese German multinational organization. We were founded in year 2000, and eventually we have evolved ourselves as a turnkey solution supplier for global pharmaceutical industry. We offer innovative solution in pharmaceutical process, in pharmaceutical packaging, ranging from primary to secondary to tertiary packaging solutions, with under the Industry 4.0 end-to-end integrated solutions, complete comprehensive system that we provide to our customers worldwide. We have direct operations in 13 nations, with more than 6,000 plus technical staff distributed among demographics to cater to our customers' requirements and support. It's a R&D driven organization again, with the research and manufacturing facilities in China as well as in Europe. So a little bit about the journey of Truking. As we said, it was foundation stone was laid in year 2000 in Changsha, China, where, just a moment, let me put the pointer on. In Changsha, China, where we have 250-acre campus for manufacturing. You can see the bird eye view over here. In 2009, the further second phase was unveiled with additional 67,000 square meters of manufacturing facilities for the pharmaceutical equipments. And in year 2014, the Truking was registered as on the stock exchange of Shenzhen stock, and the various acquisitions were made to several other process companies in China itself, making us a turnkey solution provider in sterile liquid manufacturing process. Further on, while we were making everything in-house for sterile liquid injection, including the lines, the process vessels, the water system, the packaging system, we did not had OST solution, solid dosage solution machineries. So in 2017, Truking Group has acquired the Germany-based Romaco Group, which is a prominent player in solid dosage equipment, in tablet compression, in tripod fillers, in granulation system solutions. So it actually completed our complete portfolio under a one-stop solution for either sterile liquids as well as for or solid dosage requirement from our customers. In year 2020, Truking has launched in the wake of pandemic for sure. We have launched our bioengineering solution for especially for the vaccines players worldwide, and we have catered to so many of vaccine suppliers, manufacturers, and supported them for the betterment of the humankind. So in 2021, the further expansion was done in 100% single-use bioengineering portfolio. And there was also addition in latest BFS, more advanced filling lines, etc. that was unveiled. And the launch, official launch of Truking direct offices in 13 countries was also done in year 2022. Previously, it was either with some agencies or there were unofficial working there. So that was also made in year 2022. Talking about the portfolio in aseptic fill finish solution, we specialize in high speed aseptic container filling units. either it be vials, ampoules, PFS, for bulk processing as well as for ready-to-use processing. In bulk processing, we can cater to the actual speed of 30,000 containers per hour, which can be linked with online with the with integrated lyophilization system, with the like Industry 4.0 automation, the PFS solution. Then we have a manufacturing system for the vessels, water system complete turnkey. And these are the portfolio for bioengineering, the chromatography system, the bioreactors, etc. Then further, it is downstream. When we move downstream, we have artificial intelligence-driven visual inspection system, the leak testers, and followed by the secondary and tertiary packaging systems such as cartonators, blister machine, case packers, bundler, palletizers, end of the line. So being a total turnkey solution provider, we can provide the bioengineering overall solution for the complete facility. Same goes for the natural extract medicine and general injection. Then for the intelligent BFS solution, capacity around 36,000 containers per hour from one machine. Then soft bag lines. And again, this is very popular. That is also part of the agenda today about the integration of the aseptic container vial filling lines with the lyophilizer. So the complete facility turnkey from the process support, from the design support, from the equipment support, as well as from the except the civil construction can be provided to the customers worldwide. Okay, let's come to the point about the ADC. What are ADC drugs? Antibody drug conjugates, and why the freeze drying, the lyophilizer or freeze drying is synonym. So I will be using mix of both are employed in this particular process, then what we can do in that particular area, and where the industry is struggling, and what can be a possible solution to those remedies we will be discussing in this presentation. Okay, so talking about ADC is the antibody drug conjugate. Like what is the profile of this drug, why it is used? It is basically majorly popular for the cancer treatment, and it is called as a targeted therapy drug as well, where the antibody drug conjugate are specific linkers that are used to connect the antibodies to the small molecular cytotoxic. This is very important drugs whose main component include antibodies, which are used by the linkers, and small molecular cytotoxic drugs are being deployed in the targeted cancer cell to destroy it. So the basically is this one is particularly the antibody, and the linker is used to or the drug itself, which can have a specific action, a targeted therapy against a cancer cell which is present in the body and can destroy it, and is a very specific therapy on the production as well. The major characteristic of the product as well, it being a cytotoxic in nature. So this has to be very much specific that it has to be having adverse effect on the targeted cells, whereas it has to normalize structure on the healthy cells or the good cells in our body. So once we inject the patient with an ADC drug, these are the targeted cancer cell that we want to destroy. It is internalization enable it to enter the cell. Then we have lysosomes, which contain a controlled release of it, which can target the microtubule disruption, and also or it can do the DNA strand breakage for the further destruction. So what advantages it brings to the table for the mass medicine? That it is very significant therapeutic effect has been registered with high potency, rapid action on the tumor cell, and the low risk of adverse complication on the healthy cells. Not easy to develop drug resistance. That is also very important because cancer cells is tend to known for its self evolvement against the medicine. Yeah, against the medicine it is being injected with. So ADC drug has been registered with a very a prominent effect against these cells, and also it does not easily develop the antigens against it. So the longer circulation time in the serum also ensure the larger efficacy of the medicine. Then there is less adverse effect on the non-target cells as well in this particular area. So even the ADC drugs was also developed the stage wise. So the first generation drugs, once we were developed, they were difficult to reach, and the concentration level of the cell killing was not so much prominent. Then it was also easy to cause the antimicrobial antibodies, which are in turn, neutralizing the medicine itself. Then there was lack of effectiveness. There was toxins released in the plasma, which were creating a little bit of side effects as well. In the second generation drugs, the accuracy of the target is relatively improved. Probably the extracellular release of the drug was a little bit more controlled, and there were fewer type of toxins and plasmas. Currently, we are having the third generation with us, which is having a larger stability, and its performance is improved. There is a lower coupling and shedding speed it is having. Then there is also a high drug activity, which is being registered in terms of the performance and the response it is receiving from the patients. Then antigen levels are also reduced in this particular third generation modification of the ADC drugs, which is being developed by major pharmaceutical companies. So these are the major brands that are available in the market. Currently, the patients are being, this one is treated with it. So the majorly popular is Gemzumab, made by Pfizer, which is used for the leukemia medicine. Then uh, we have a Polivy made by Roche, which is for the lymphatic carcinoma. And then there is a Blenrep, also a major trade name made by GSK for the bone marrow cancer. So this is a complete list of being these complicated medicines are being used for.
Sorry, Rakesh, you muted yourself, I think.
I've been muted myself from quite a long time, or
No, no, no,
No,
no,
no,
no.
just few
Just
seconds.
yes.
okay. Oh my goodness, how it's happened? I don't know. Maybe okay. Adolf
It's
B.
okay, it's
Like
okay.
I think something been done there. So I'll go back a little bit. I think you guys missed me here in this slide.
Yeah.
Yes.
All right. So what I was discussing that we have these drugs. Just a moment, let me put the pointer. So we have these drugs with the big pharma that is already in the market, and the patients are being treated on these medicines. The majority of the drug is MABs family, first of all, and this one is Gemdizumab made by Pfizer and being used for treatment of leukemia. And then there are other drugs being listed with the brand name, with the medicine name, majorly of the MABs family that is being employed in ADC. And then there is these are like, for example, Blenrep is a major trade name for the GSK, which is used for the bone marrow cancer and others. And these are the FDA approval dates, and these are like very much in the market already. So the furthermore is like being a more specialized adaptation in the market. This ADC drugs is bound for an exponential growth as well. For example, this Tatsumab is projected to be a value of 6.2 billion US dollar by year 2026, followed by the Enfortumab with 3.5 billion US dollar, uh, which is followed by the Vibutumab for the 1.8 billion US dollar, is a forecast by 2026. So the growth and the and the product value and the medicine cost, as well as is a huge a huge potential for the big pharmaceutical organization to manufacture and expand in that particular area as well. Okay, so once we were having these drugs as a liquid injection in various containers, such as the vial, such as the PFS as well. So why we require freeze drying of these equipments? First major advantage that FDA and also our users have recognized that there is ease of processing liquids. which is more aseptic in nature as well as compared to the handling a powder or other formal dose of the equipments, the dosage. Then there is second major advantage that freeze drying lyophilization brings to the table is a prolonged shelf life of the product without reducing its efficacy. So that increase the yeah. You guys can hear me still, right? So that increase the shelf life of the product itself, causing minimum recalls and back to the brand incidents. Then it is like known for it is also proven in general injection as well as in ADC drugs that it provide a greater chemical stability of the product once you freeze dry it, convert the liquid injection into a dry powder injection using a freeze dryer as compared to the only liquid injection going to directly to the market for example, if it may have the shelf life of 2-3 weeks, it can be changed to 2-3 months in case of ADC drugs while freeze drying the product. So the complete process flow. Sorry, just to be sure, I'm audible, right? Yeah, I'm audible. Fine. Okay, so talking about the overall process of the containers handling as well as the freeze drying process that we can cater in overall. So first of all, the user select the right containers. In this case, is we are targeting the vials. We have the washing machine. First, we for the bulk processing we are talking about. We have a washing machine to wash the vials up to log 3 reduction of endotoxin. Then we sterilize the same container to achieve log 3 or log 6 endotoxin reduction. Then we have entered the filling machine where we dose the containers with the desired set of dose in the container using a filling machine. Then we half stopper it. Then under the aseptic conditions, it is being transferred to a freeze dryer, which can be automatically loading the containers row by row or using an AGV inside the machine. And after that, the lyophilization has a very majorly we can target it as a 3-step. First is the freezing, then we have primary drying, then we have secondary drying, where the sublimation and desorption happens, and the liquid directly transfer itself into the powder, dry powder. Then the freeze drying process is complete. In this particular stage, we have a powder here. Then there is inside the lyophilizer only using hydraulics. We do the full stoppering operation, and the containers are row by row automatically come out of the freeze drying machine, which can further be connected to the aluminum crimping capping machine, which can follow by the visual inspection machine, which can follow by the labeling machine, cartoning machine, case packer, the stacker, the case packer, the track and trace, everything. So this complete end of the line solution can be delivered for the ADC as well as for general injection, employing vial lines, aseptic fill finish lines, lyophilizers, the packaging systems, complete streamlined under high capacity as well. So lyophilizer talking per se, that first of all, this comes in majorly 3 sizes, 3 scales, I would say better. So first one is the lab scale, where the scientist develop the medicine recipes. He developed the procedures, the parameters, the vacuum, the temperatures, etc., the solvent concentration. All has been developed in this particular lab scale lyo in their microbiology labs. Then the same parameters can be replicated in the pilot scale, where we have a very lower capacity, but still you can file your batches. You can have a trial batches, etc. Even you have production batches if you have a lower batch size, considering the very expensive product ADC medicine is. And third one is the production scale machines. With this size, this can be from 1 square meter to 50 square meter machine, depending upon the batch size and the requirement of the user. Again, you can either extrapolate directly from the lab scale to in here, or you can follow the procedure from lab scale to the pilot scale, then to the production scale, depending upon the expansion, the batch size, and the calculations. These are the production scale freeze dryers, lyophilizers, which can adapt to the layout constraints of the user. For example, some facilities are greenfield, some are brownfield. Majorly from the brownfield, we have a challenge that constrain layout sometimes. So the machine can adapt to the different design. Like for example, this is a two-floor setup with the chamber on the top, the condenser, and the refrigeration kit on the bottom. This can be with the standard orientation with the vertical condenser. We can put it on the side. We can put multiple lyophilizers side by side, depending on the batch size upon the user requirement again. So we can, under Industry 4.0, as we said, we can transfer the containers under varied level of containment, starting from OEB level 3 to OEB level 5, which is isolator for cytotoxic product, talking about ADC medicine precisely. It can automatically load the freeze dryer with the Rovero loading of the containers inside, and offloading is also done automatically Robero, or you can have an automatic guided vehicle where you have one wire line, particularly here in this video, and you have multiple lyophilizer 1, 2, 3, and you have this automatically guiding vehicle that is transporting the vial from this particular area to this particular lyophilizer loading. This card is also can be incorporated with the open wraps OEB level 3 level of containment. If more strictness is required, then we have to go for this fixed type of solution in this particular point. So talking about the industry bottleneck and the pain points, first of all is that the ADC medicine per se is very expensive, and the batch size, the raw materials, the load of the plant is also very crucial. So it is very important to choose the right equipment, and the equipment which is used to process the liquid containers to the lyophilized container must be capable enough to have a better impact in terms of the performance and the freeze drying parameters, how you contain the system itself. The first one is for the equipment reliability. The second one is also very important. Being endotoxic in nature, it can be dangerous for the operator and the environment. So we had to ensure certain measures that ensure the safety of the personnel who is operating the equipment. And then we have regulatory guidelines as well that need to be followed in to ensure the batch uh, efficacy per se and sterility overall process in the manufacturing process that is being incorporated. So first of all is that freeze drying is a very specific equipment. Being a stationary machine is basically running the process. It has to be capable enough to withhold the strict parameters of the CGMP practices and FDA guidelines. Per se, talking about our machine, we have a very special and unique in-house manufacturing of all the critical components under one roof for the freeze dryer. The main heart of the product again is the shelf, where we put the containers automatically inside the freeze dryer. For example, these are the shelf. We call it shelves, which can have in a machine it can have 5, 6, 7, 11 shelves stacked using a hydraulic, depending on the machine size. First of all is that while making the shelf, we use vacuum brazing technology, and is a fully robotic process where we ensure there is a proper welding. Also ensure there is a practically zero thermally dead area in the shelf, so that there is maximum thermal conduction, and eventually you have a better cycle time and a more efficient machine running in your plant. So I have a just small video about how we process our shelves in our fully automatic robotic welding process. So this is how the shelves are being processed and automatically welded using robots in our factory. If you see basically the brazing welding technology is a very comprehensive process and require lots and of investment in terms of manpower and equipment. And in the end, the shelves, which is made only with 18 mm thickness, is much more have much more tensile strength, and have a better performance as compared to the traditional spot welding shelves that are available in the industry with that has some constraint drawbacks as well. So this is the further process for the ensuring the welding. Basically, this particular point shows how we safeguard, how we ensure the equipment is designed and is made and is built to last under the toughest vacuum condition that machine generates, and also achieve industry's best leakage rate in the machine of 0.01 mbar liter per second. The shelf is now complete. Then further, after this welding process, it goes to an oven where they keep it for 2 weeks to ensure the proper leakage and linkage. Sorry, and the process of shelf making is then complete. Okay, so I'll move to next. Okay. Second major part of the machine is the chamber. So how we ensure there is once the machine, the chamber of the lyophilizer freeze dryer is fabricated, it must not have any type of leakages or welding leakage. So at Truking, what we do is being in-house manufacturing, there is only one point to weld for the whole chamber made in one piece, and that ensure give us a better performance in terms of the leakage processing as well. Sealing surfaces and the sealing groove shall also be processed and formed at one time after the welding process. So these kind of fully automatic processing technique can largely increase largely reduce, I'm sorry, the risk of product failure while you process a very expensive ADC drugs or any other cytotoxic product in the freeze dryer. The machine has to be capable for that particular point. Then talking about the hoses, again, these are the hoses which are used to transfer the silicone oil from the circulation pumps to the shelves of the lyophilizer, which is used to transfer the heat or the heating to the shelves, which is a major part of the lyophilization process. These shelves are being tested for 100,000 plus workload, and it has a very high performance as well in this particular point. Then talking about the condenser being a helical coil, this is the third part of the lyophilizer prior to the refrigeration skid. The job of the condenser is to ensure the surface area uh, for ice trapping. When we evaporate the solvent from the product, it has to be being trapped somewhere. So the vacuum pump, what it does is it compresses the refrigerant, and the refrigerant when enter the condenser, it is encountered with the larger surface area, and gases expand quickly and causes a minus 75 degrees temperature in the condenser. And once the isolation wall between chamber and condenser is open, the solvent vapor are directly being trapped inside the condenser, and the cycle is being processed further, and you have a lyophilized product in the chamber. So again, like from the equipment reliability point of view, we have already discussed about the workmanship that is required to make these kind of machines. Then further in the refrigeration and in the vacuum skid, there is also we need to be having high quality pumps and compressor that is required to be used in the particular point. So in the refrigeration, the pressure monitoring and differential protection is being achieved by the either user can choose between the reciprocating compressor from Bitzer, or they can have a dual stage screw compressor from Micom. Then they also have overheating protection, overload protection, and other multiple protection for in case of the failure rate. Then there is low temperature and abnormal protection, more than 30% of the redundant cooling capacity, and the compressor in the late freezing cycle can be support to complete the whole freeze drying process. Basically, we can also add the redundancy in the compressor, like the once the primary drying is started, the load between the compressor can be divided if you have multiple machines, compressor skids available that ensure the lower compressor failure rate, as well as you have a faster reaction in terms of the performance of the equipment. Again, second, in the vacuum pumps, we have a vacuum pump, then we have roots pump to create the vacuum. As I said before, we need to have trap the ice in the condenser of the solvent. So that need to be a principle from can be again one use and one standby, as a redundancy can be employed in the whole system to ensure the equipment is reliable and you have a more safety at your disposal once you are processing the medicine. Same goes for the circulating pumps as well, which can be again with the redundancy, as well as we have as per standard only the design and the model choosing of these equipments are as per already considered the over capacity for the better usage. Second, again, the major point about the documentation from the equipment reliability point of view, the system is equipped with SCADA software 21 CFR Part 11 compliant with the audit trail. Then it has a flexible automatic and manual operation mode that is also sometimes used while maintenance or some part of the product recipe providing. Then electronic data is stored in a separate controlled computer, which enable data indexing, export online and offline backup and transfer. Then automatic lyophilized recipe and flexible recipe modification function is also provided. For example, this is the documentation, all the mimic and everything you can have an audit trail. Then there is complete PNID mimic that is can be used in SCADA software by the operator to see which valve is open, which compressor is working, which vacuum pump is working, at what stage, what is the temperature of the chamber, what is temperature of condenser. All these parameter can be seen in a very interactive way that that also ensure the equipment is reliable enough to have a better process control. That is very much critical. Okay, so we have talked about the equipment's role in ensuring the ADC product or some specific product is safe. So second thing, the major bottleneck of industry is how to ensure the safety of the product as and the operator itself, the human point in this particular here. So we have toxic substance of ADC drugs. Then it can be a DNA damaging agent. It can be microtubule inhibitor. DNA transcript and inhibitor is also available, is also present in this drugs itself. Just a moment. How we safeguard the environment? How we safeguard there is no dangerous vapors or the solvent has not been released anywhere in the area which can be dangerous to the operator, to the working staff. So we can, first of all, we can use a dry type vacuum pump. Second, it can also use a BIBO. We will discuss these points, and then it can be toxicity treatment during the production process is also necessary. Then toxic treatment during the maintenance is also very important while we have equipment working on the ADC drugs. So first of all is the safeguard measures for operator and the equipment. So to ensure the stable operation, the vacuum pumps and the root pumps, the configuration follow principle of redundancy, as we said before. Then we can have a better vacuum control as well. These are the like either user can use an MKS vacuum manometer or a Pirani vacuum manometer to see the better control over the pressure parameters in the machine during the cycle is running. So this also gives us a lot of data to see if something is going to get wrong, or we can control the batch. We can save the batch being seen of multi-million-dollar batch at a time. So these points are very much important as well. So in dry product evacuation, this is the data that is being mentioned. Then vacuum pump adopts advert type, dry type to avoid step to replacing oil type. So while we're processing a dangerous cytotoxic product, so user usually prefer a dry type vacuum pump over an oil type because oil type can somehow get contaminated with the solvent. And in this case, please note that solvent is a cytotoxic, is a contagious substance. So we can adopt for a dry type pump in this particular area, and there are more remedies that we will discuss later in this. So second is that for the operator safety point of view, we can use Vivo box in, box out filters that are being installed at the critical parameters where there is a possibility of release of toxic substance to the environment from the machine while processing the batch, or even after the CIP/SIP of the system while the batch has ended. So hazardous waste gas is collected and filtered in these BIBOs, then filter back in and out can be done and directly go to the discard. So BIBO treatment can be 2 times on the vacuum system. First of all, vacuum system is part of the lyophilizer. And this is the BIBO, put it here. This is the vacuum pump. This is in a PNID. This is a condenser isolation valve chamber of a lyophilizer controlled by hydraulic. So what we do is that at the inlet of the vacuum pump, we can add a BIBO, which can be acting as a barrier for anything contagious coming out into the system. And same process can be updated at the outfeed of the vacuum system as well. So there are BIBOs at two critical locations that ensure there is no suction, no solvent, no traces of vapors that is being harmful is being released to the area, to the room. Okay, so then further we have more measures to ensure there is on the vacuum pump system, there is no containment, no contagious substance coming to the area. So first step is that this is the two PNIDs you can see. One is before, one is after the vacuum pump. And then first of all is that we can stop and close the inlet and exit valves of the vacuum pump and the roots pump before the cleaning, because now we are into the CIP/SIP stage. CIP stage of this particular system. So we can close, this is vacuum pump 1, vacuum pump 2, we can close this valve, we can close this valve. And then in the second step, we can clean the inlet pipe of the vacuum pump automatically through CIP/SIP lines. These valves, please note, is closed. The loop is cleaning the complete lines, these one, and the water is going to the drain. In the third step, that the cleaning of the vacuum pump outlet point. So this is in the outlet also. After closing these valves, we can have a CIP done online on these particular pipes. And in the fourth step, we can because being these valves are closed, we can dismantle these pipelines in the form of a major maintenance. Dismantle these lines in outfeed, these line is infeed of the pumps, and it can be taken to a certain area which can be go for the further maintenance to ensure that there is no contagious substance being released in the classified area in the pharmaceutical factory. So they can be taken to a specific area in the factory, which can be overall sanitized after the cleaning process is done. So this was previously we were discussing about the vacuum, how to ensure, how to make SOPs, how to make ensure the safety of the environment and the product linked with this equipment and the contagious substance, the product contact parts that are being used in the lyophilizer freeze dryer machine. Now we talk about little bit about the filter integrity testing. and under which regulation it states what steps need to be followed. So in order to comply to the latest pharmaceutical guidelines for ADC drugs, the machine need to be adapted to higher safety protocols. As per the EC Guide to GMP for Sterile Medicinal Products 1997 edition, it clearly states that the integrity of the sterilized filter should be verified before use and should be confirmed immediately after use by an appropriate method, such as bubble point, diffusive flow, or pressure hold test. Under the FDA guidelines in 1987, the normally it state that integrity testing of the filter is performed after the filter unit is assembled and sterilized prior to use on the equipment. So this is very important. So here we have a regulation guideline for the online filter integrity testing procedures. And again in the PDI report, it also states that generally is regarded as a CGMP practice that filter or the filter system routinely be integrated tested both prior and after use. So prior and after use is very important. So filter integrity test under regulation, we can have like a certain set of a procedure named as the water intrusion method. On the left side, you can see the equipment which is being connected to the filter with the housing in line. It has a particular valve for exhaust. It has power supply lines, the equipment. Then these are the air inlet line, then air outlet line. The air compression source is connected to the device which is using intrusion test. And this is the inlet and outlet sockets of the tester itself. So what is the principle that being a hydrophobic membrane in nature of the filter that is being used, it resists water. So the smaller the pore diameter, the greater the pressure required to squeeze the water into the hydrophobic membrane. So it's very obvious. So therefore, under the certain pressure, we measured the water flow squeezed into the filter membrane to judge the pore diameter on the filter element. So that is how the principle basically of water intrusion works. Then in the method that but how we do it, we fill the water, fill the filter inlet with the test water until it overflows, first point, and then we stop the inflow, second point. Then we keep the test port under the test pressure through a tester, such kind of equipment, and judge the integrity of the filter through the water flow within a specified time test. So it's linked with the water flow and the time that is stipulated on the device. We make a recipe on this while the qualification. Then integrity detector test within test scheme can be employed. Either within test or water intrusion test can be done. So we can adhere to the WIT water intrusion test protocols in this particular scenario to ensure the performance is adequate, and we can prove the integrity in our documents at the design stage. So what is the flow rate of under the regulation states for the filter integrity? First, we install the tester, then filter is wetted, then we connect the tester, then tester boot self and first self test itself. Then there is a new program running test being linked with the steps that we have mentioned in this particular point. So how it is done? So integrity testing method can be offline and online. Under online, it can be automatically online detection. Then there is manual online detection also available. Under automatic, we have integrity detector that we have seen. Then there is a within test that we'll also discuss later in the slide. So first of all, that filter integrity test under regulation, but its state is that in method number one, we have such kind of equipment available in industry. For example, in this Mimic, we have shown a Poltronic flow mat instrument that is used for integrity testing. This is a filter. We have a processing, a valve, then we enter the filter, then this is the outlet of the filter. We have a filter vent, which is connected to the device in here. And we have this outlet as well, goes connected to the further drain. It can also be these some devices also comes with a remote working for the controlling the integrity testing equipment. So integrity testing establish a communication with PLC operation, and reports are perfectly integrated with the SCADA that can be further linked with the auditorial and your process requirement for the point. And method two is using the within test. water immersion test method. So in this particular point, we have a very specific set of protocols in this domain. That the volume change is like is in a high school we have learned a formula P1 P naught V naught is equal to P1 V1. That is a basic governing formula of this within test. So the volume change is obtained by measuring the corresponding pressure drop basically. And the rate of volume change is equivalent to the flow rate. So volume change, flow rate. If there is a deviation, there is a issue in integrity of the filter. So these are the lines that are being used for the pressure, compressed air, pure steam being connected to the filters. And that is how we are conducting the test particularly in this particular area. We'll also have more slides for the within test at later stage. So first of all is that the detection process, let's talk about it, which is common for both. And then the complete preparation of water for filter injection through the steam condensing equipment is done. The selection filter to be testing, control and open the automatic diaphragm valves, fill the preparation water through the water injection nozzles until it is full. And then we have the tester judges integrity of the filter through the water flow in the specified time that we can provide on this. Okay. Okay, so second one is I think we are also having some questions, so we'll take it at the later stage on this one. In terms of automatic phase time loading the loading system and inspection. Okay,
Yeah.
so
Yeah,
we'll come to it.
No,
Yes.
Rakesh, like the idea is when you finish, we will follow up all these questions, okay, and then we can give option to guys to make any question, okay?
Perfect. Okay. So we have pressure probes in this in the talking about the automatic integrity detection in the within test. We have complete preparation of water for filter injection through the steam condensing equipment that is being employed for the integrity tester in this particular domain. Then we select the filter to be tested. Control and open automatically diaphragm valve, fill the preparation water through the water injection and nozzle until it's full. Then inflation is done through the compressed air at 2.5 bars. Within the specified time, as we said, the within test is linked with P naught V naught P1 V1 formula that is being employed within the specified time, and pressure and decay is very important in this particular domain. And the temperature is corrected by the temperature probes with high precision. Pressure change at a pressure probe of 0.02% accuracy, which is calculated by the computer itself, which is coming from the device. After that, we can know the integrity of the filter element as a final result with us, which can be used before and after the batch has completed. This method can be suitable for integrity testing after sterilization. I'm sorry, I missed this. So this one is basically we done after sterilization, the filter has been put in place. So after this manual and online automatic integrity detection, we can have a kind of comparison between the water intrusion and the within test. So on this side, we have a water intrusion, this side a within test. Both of these are being employed for the filter integrity testing that is being specified, which is required and incumbent process for the manufacturing readiness of the equipment. So applicable regulations are domestic GMP and FDA. It can comply to that. Within is not so much popular in the FDA guidelines. Then soaking water requirement, we have purified water, WFI, inline steam condensation preparation as a utilities being used. Then this is online steam condensation preparation suitable for UGMP guidelines. Then we have a number of detectors. There is one detector, 1 to 1. One is for the detector, one is for the freeze dryer. So sorry. Then high precision pressure and temperature probe is used in within test. Then batch data record, PLC and recorder communication included in the batch data recording is done in the WIT test. Then PLC and recording communication included in the batch data recording, almost similar in this. Then talking about the convenience of operation between the water intrusion and the within test. So there is operating strength is not like we don't need a very specific operators for conducting a water intrusion test, and it is also very good overall good stability and safety that it can ensure and being accepted by the regulatory guidelines as well. In case of within the operators knowledge is also of the similar level, and stability and the safety is also being accepted in the as per the guidelines stated by the regulatory in this. Okay. So basically, this is we thanks everyone for the input for the being a listener to us. So we can have a question and also on this now if some audience requested.
Sure. Thank you very much, Rakesh. It was very insightful presentation. We have some questions here in the chat. Maybe you can you read them by yourself? Okay.
So I think let me open.
Yes, here, yeah.
Okay, so sir, first of all, we'll take questions for AI-driven inspection system works for plastic poly translucent vial. Sir, like in case of the containers, it is also very popular these days. We have polyurethane, polypropylene, sorry, material of containers being employed instead of the glass containers, because it has been observed in some studies that there is a microflaking that can occur inside the vial containers that can be reactive to some of the drugs. So in case of your vial is visible, is translucent, we can see the product. It is not like a completely opaque container, or it does not have some completely opaque liquid like milk-like or a blood red like ferrous sulfate. So if that is not the case, plastic containers can be inspected in a visual inspection machine. In case of, sir, like you have fully opaque container or fully opaque liquid medicine, you had to go for an X-ray inspection. So we also have X-ray inspection where we can inspect for the foreign object such as a rubber plug or a metal particle, a big debris that is being accumulated in the container. So that can be done. So I hope I answer your question. So that is we can do it. Then what are the major challenges in tech transfer that is from development scale to the production scale? Okay, so basically that from the development scale can be either a lab scale or it can be a pilot scale equipment. Majorly, it will be a lab scale. So the same recipe, if you're using a similar equipment in the lab as well as in the production, the parameters, most of them will be the same between the laboratory and the production. Only difference it might have will be like the production scale has an online CIP, SIP, high pressure of the water and CIP cleaning mechanism, whereas in lab scale, you had to clean manually because it being a very small machine. So if we are following those parameters, there is not a challenge. Being with the same recipe that can be extrapolated from, for example, 5 liter to 5,000 liter batch size can be done easily. Okay, so I think these are majorly questions that we are having. So for ADC product, what is the market requesting equipment supplier in terms of automation, freeze dryer, loading alone? So Mr. Adolfo, this particular question is that being ADC is a very specific medicine, the batch size is very expensive as well. The users have two type of solution. One, they can go for a bulk solution where their batch size are comparably higher, bigger. So in that particular case, you need a fully automatic, complete aseptic fill finish line, vial line, which contain washer, sterilizing tunnel, filling. Then there will be an automatic loading to the freeze dryer. Then there is a capping machine. In this particular scenario, your output is high if you have a larger batch size of the product. In second scenario, in ADC product, because being sometimes the batch size are not higher, being the product is also very expensive, people use ready-to-use application machines as well. In this, they use pre-sterilized containers, which can be directly loaded in the filling machine, which is either under open wraps, either under isolators, depending on the level of containment required. Majorly, isolator will be required. That is a major demand coming up, if I rightly answer your question, which can be automatically loaded to the lyophilizer, which is inline, then followed by the capping, followed by the secondary, tertiary packaging systems. So that can be done. In ADC, we can see that the demand of RTU is high, ready-to-use containers is high, and being the batch size little bit limited, and they need equipment which are having a huge safety protocols and over redundancies as well in this particular area. So I think I hope answer your question, sir. Time difference between lab and commercial scale. Time difference, Rajiv sir, if you mention about the manufacturing time, if you're mentioning, we can say that around 6 months FET ready for the production scale, and we can say about 4 months for FET ready for the lab scale lyophilizer. So, okay, what is the typical batch size of ADC products? So, Carolina, it's very specific question. I'm not the right person to answer this. It's very subjective as well. It depends upon the product. Basically, what is the timeline for a batch size? So it depends upon the product, the solvent concentration. So we need to have a scientist and from the process side to answer this particular question. So it's very subjective. It depends upon the product, which is either it is a Tazumab, either which map size it is coming from. So it's very subjective. So anything else?
I think there was a question from Ulan Shu. Does Trucking can provide ADC drug preparation, ultrafiltration, etc. equipment?
Okay, so like for the ADC drug preparation, Truking has a complete suite that we can provide in bioprocess equipment. We can have complete bioengineering portfolio that Truking can supply to you. It can, like, from the bioreactors to fermenters, from the cell line culture and expansion. It starts from that particular area till the chromatography and the ultrafiltration. All the equipment on turnkey basis can be supplied by Truking. Once we make the product through these equipment, we can also supply the primary packaging machine, which is a aseptic fill finish line, lyophilizer, inspection machines, packaging systems, end of line. So you be assured for the complete solution that can be delivered to you in of a very high quality under record time from one single supplier, making the design and manufacturing in-house from the manufacturing facilities in China as well as in Europe.
Okay, so is there any question more? Any of you? This is your chance to ask Rakesh.
So if there is not any questions, you can reach us. We can drop our email IDs to Mr. Adolfo, and any questions or any process-related question, you can ask us and we can give you a solution. So one more question is that any aspect for equipment can be customized. So yes, Jeffrey. So the equipment actually in today's industry in aseptic container handling, everything is 100% customized. It is customized based upon your requirement. It is customized based upon your plant layout. Either it is a greenfield, brownfield expansion. What is your process requirement? You are making which type of medicine? You require which type of containment? You require which type of precautions and procedures? So it's 100% customized. So like you can state the requirement, we can have a in-depth discussion. Our scientist and our in-house process expert will guide you on each and every step.
Okay, so well, I think that's well. One more question.
Any pharmaceutical company in EU using to yes, there's a lot of company, Mr. Rajiv, in EU. I don't know in which country you are from, sir, but in European Union, we have several installations. We can give you a list. You please reach out to us about the present installations. Or if you're mentioning about any installations of our equipment on EU GMP plants, we have lots and lots of it, even in US FDA. Any pharmaceutical company in Cyprus? Yes, yes, yes. I heard. I need to check. I need to get back with my team. Please give your details. Even I think you can drop your email ID in this chat itself. I will reach out to you about the details about the installation in Cyprus. We can even call you, and we can. This is also open invitation for all the attendees to come visit Truking facility. See, because seeing is believing, and you can have a first-hand experience on the bioprocess equipments, on the packaging equipments, and then how we do it, how we make it, and what is the scale of production that we do it in process. Either you can come to our China factory, or you can come to our factories in Italy, in Germany, that we can have has a description there. Also, we are exhibiting in Akima, which is just a couple of months away. We can have a meeting over there as well. We got your email ID, Mr. Rajiv. Will be in touch.
Brilliant, Rakesh. Thank you very much. In the name, I think, of all the guests, thank you for your time and for your nice presentation. And we'll keep in touch.
Thank you very much.